Hi and welcome to the Mechanics of Healing. This is part 1K and about the nervous system, which is video 3 on the nervous system and the central nervous system and brain. This video includes how and what synapses are and how they work, as well as the central nervous system and the brain. When we do the course on grid keeping in the future, we will see the similarities between the nervous system and brain and the Earth's grids and the universal mind. The reason we are doing these videos on anatomy and physiology is because we will find many similarities between how the Earth works and how our bodies work. In the course on wholeness and holism, we learnt about the universal design template and how the universe is a fractal whole, with each part connected to and reflected in the whole. We will find out more about this later in the Mechanics of Healing. This is a crash course in knowing your body, its anatomy and physiology. So enjoy the following video from Crash Course. There are 2 videos in this topic. Thank you for watching. Be sure to watch the next part, part 1L.
What's 1,000 times thinner than a piece of paper, more numerous in you than grains of sand on a beach, and proof that the smallest things can sometimes be the most powerful? I'm talking about the synapse, the meeting point between 2 neurons. If your neurons form the structure of your nervous system, then your synapses, the tiny communication links between them, are what turn that structure into an actual system. Because as great and powerful as your neurons are, when it comes down to it, their strength and their purpose lies in their connections. A single neuron in isolation might as well not exist if it doesn't have someone to listen to or talk to. The word synapse comes from the Greek for to clasp or join. It's basically a junction or a crossroads. When an action potential, and if you don't know what an action potential is, watch the last episode, sends an electrical message to the end of an axon, that message hits a synapse that then translates or converts it into a different type of signal and flings it over to another neuron. These connections are rather amazing feats of bioelectrical engineering, and they are also ridiculously mind-bogglingly numerous. Consider that the human brain alone has 100 billion neurons, and each of those has 1,000 to 10,000 synapses. So you got somewhere between 100 to 1,000 trillion synapses in your brain. Each one of these hundreds of trillions of synapses is like a tiny computer all of its own, not only capable of running loads of different programs simultaneously, but also able to change and adapt in response to neuron firing patterns and either strengthen or weaken over time depending on how much they're used. Synapses are what allow you to learn and remember. They're also the root of many psychiatric disorders, and they're basically why illicit drugs and addictions to them exist. Pretty much everything in your experience, from euphoria to hunger to desire to fuzziness to confusion to boredom, is communicated by way of these signals sent by your body's own electrochemical messaging system. Hopefully, you know enough about email and texting etiquette to know that if you're gonna communicate effectively, you have to respect the sanctity of the group list. It's not a great idea to send a mass text to all of your friends first thing in the morning to give them the urgent news that you just ate a really delicious maple bacon donut. Seriously, people, if you happen to have a friend who truly adores bacon, then an email would suffice. But if you're out clubbing and suddenly Bill Murray shows up and starts doing karaoke, then that would be a totally appropriate time to notify all of your friends at once that something awesome is happening, and they better be a part of it. And in much the same way, kind of the same way, your nerve cells have 2 main settings for communicating with each other, depending on how fast the news needs to travel. Some of your synapses are electrical. That would be like an immediate group text. Others are chemical synapses. They take more time to be received and read, but they're used more often and are much easier to control, sending signals to only certain recipients. Fortunately, your nervous system has better text etiquette than your mom and knows when each kind is appropriate to use and how to do it. Your super super-fast electrical synapses send an ion current flowing directly from the cytoplasm of one nerve cell to another through small windows called gap junctions. They're super fast because the signal is never converted from its pure electrical state to any other kind of signal the way it is in a chemical synapse. Instead, one cell and one synapse can trigger thousands of other cells that can all act in synchrony. Something similar happens in the muscle cells of your heart, where speed and team effort between cells is crucial. This seems like a good system, so why aren't all of our synapses electrical? It's largely a matter of control. With such a direct connection between cells, an action potential in one neuron will generate an action potential in the other cells across the synapse. That's great in places like your heart, because you definitely don't want half a heartbeat. But if every synapse in your body activated all of the neurons around it, your nervous system would basically always be in group text mode, with every muscle fiber and bit of organ tissue always being stimulated and then replying all to the whole group, which would stimulate them even more until everyone just got maxed out and exhausted and turned off their phones for good, which would be death. So that would be bad, which is partly why we have chemical synapses. They are much more abundant, but also slower, and they're more precise and selective in what messages they send where. Rather than raw electricity, these synapses use neurotransmitters, or chemical signals that diffuse across a synaptic gap to deliver their message. The main advantage chemical synapses have over electrical ones is that they can effectively convert the signal in steps, from electrical to chemical back to electrical, which allows for different ways to control that impulse. At the synapse, that signal can be modified, amplified, inhibited, or split, either immediately or over longer periods of time. This setup has two principal parts. The cell that's sending the signal is the presynaptic neuron, and it transmits through a knob-like structure called the presynaptic terminal, usually the axon terminal. This terminal holds a whole bunch of tiny synaptic vesicle sacs, each loaded with thousands of molecules of a given neurotransmitter. The receiving cell, meanwhile, is, yes, thankfully, the postsynaptic neuron, and it accepts the neurotransmitters in its receptor region, which is usually on the dendrite, or just on the cell body itself. And these two neurons communicate even though they never actually touch. Instead, there's a tiny gap called a synaptic cleft between them, less than 5/millionths of a centimeter apart. One thing to remember is that messages that travel via chemical synapses are technically not transmitted directly between neurons like they are in electrical synapses. Instead, there's a whole chemical event that involves the release, diffusion, and reception of neurotransmitters in order to transmit signals. And this all happens in a specific and important chain of events. When an action potential races along the axon of a neuron, activating sodium and potassium channels in a wave, it eventually comes down to the presynaptic terminal and activates the voltage-gated calcium channels there to open and release the calcium into the neuron's cytoplasm. This flow of positively charged calcium ions causes all those tiny synaptic vesicles to fuse with the cell membrane and purge their chemical messengers. And it's these neurotransmitters that act like couriers, diffusing across the synaptic gap and binding to receptor sites on the postsynaptic neuron. So the first neuron has managed to convert the electrical signal into a chemical one, but in order for it to become an action potential again in the receiving neuron, it has to be converted back to electrical. And that happens once a neurotransmitter binds to a receptor, because that's what causes the ion channels to open. And depending on which particular neurotransmitter binds to which receptor, the neuron might either get excited or inhibited. The neurotransmitter tells it what to do. Excitatory neurotransmitters depolarize the postsynaptic neuron by making the inside of it more positive and bringing it closer to its action potential threshold, making it more likely to fire that message onto the next neuron. But an inhibitory neurotransmitter hyperpolarizes the postsynaptic neuron by making the inside more negative, driving its charge down, away from its threshold. So not only does the message not get passed along, it's now even harder to excite that portion of the neuron. Keep in mind here, any region of a single neuron may have hundreds of synapses, each with different inhibitory or excitatory neurotransmitters. So the likelihood of that postsynaptic neuron developing an action potential depends on the sum of all of the excitations and inhibitions in that area. Now we have over 100 different kinds of naturally occurring neurotransmitters in our bodies that serve different functions. They help us move around and keep our vital organs humming along, amp us up, calm us down, make us hungry, sleepy, or more alert, or simply just make us feel good. But neurotransmitters don't stay bonded to their receptors for more than a few milliseconds. After they deliver their message, they just sort of pop back out and then either degrade or get recycled. Some kinds diffuse back across the synapse and are immediately reabsorbed by the sending neuron in a process called reuptake. Others are broken down by enzymes in the synaptic cleft or sent away from the synapse by diffusion. And this mechanism is what many drugs, both legal and illegal, so successfully exploit in order to create their desired effects. These drugs can either excite or inhibit the production, release, and reuptake of neurotransmitters, and sometimes they can can simply mimic neurotransmitters, tricking the neuron into thinking it's getting a natural chemical signal when really it's anything but. Take cocaine, for example. Don't take cocaine. Once it hits your bloodstream, it targets three major neurotransmitters: serotonin, dopamine, and norepinephrine. Serotonin is mainly inhibitory and plays an important role in regulating mood, appetite, circadian rhythm, and sleep. Some antidepressants can help stabilize moods by stabilizing serotonin levels. And when you engage in pleasurable activities like hugging a loved one or having sex or eating a really great donut, your brain releases dopamine, which influences emotion and attention, but mostly just makes you feel awesome. Finally, norepinephrine amps you up by triggering your fight or flight response, increasing your heart rate, and priming muscles to engage, while an undersupply of the chemical can depress a mood. So in a normal, sober state, you got all these neurotransmitters doing their thing in your body, but once they've delivered their chemical payloads, they're usually diffused right back out across the synapse to be absorbed by the neuron that sent them. But cocaine blocks that reuptake, especially of dopamine, allowing these powerful chemicals to float around and accumulate, making the user feel euphoric for a time, but also paranoid and jittery. And because you have a limited supply of these neurotransmitters, and your body needs time to brew more, flooding your synapses like this eventually depletes your supply, making you feel terrible in a number of ways. Cocaine and other drugs that target neurotransmitters trick the brain, and after prolonged use may eventually cause it to adapt, as all those synapses remember how great those extra chemicals feel. As a result, you actually start to lose receptors, so it takes even more dopamine and finally cocaine to function normally. Sometimes the best way to understand how your body works is to look at how things can go wrong, and when it comes to your synapses, that, my friends, is what wrong looks like. In their natural healthy state, your synapses know when to excite, when to inhibit, when to use electricity, and when to dispatch the chemical messengers. Basically, a healthy nervous system has the etiquette of electrical messaging down to, well, a science. Today you learned how electrical synapses use ion currents over gap junctions to transmit neurological signals, and how chemical synapses turn electrical signals into chemical ones using neurotransmitters before converting them back to electrical signals again. And you learned how cocaine is a sterling example of how artificial imbalances in this electrochemical system can lead to dysfunctions of all kinds. This episode of Crash Course was brought to you by Logan Sanders from Branson, Missouri, and Dr. Linnea Boyev, whose YouTube channel you can check out in the description below. Thank you to Logan and Dr. Boyev for supporting Crash Course and free education. Thank you to everyone who's watching, but especially to our subbable subscribers like Logan and Dr. Boyev who make Crash Course possible. To find out how you can become a supporter, just go to subbable.com. This episode was written by Kathleen Yale, the script was edited by Blake DiPestino, and our consultant is Dr. Brandon Jackson. It was directed by Nicholas Jenkins and Michael Aranda, and our graphics team is Thought Cafe. James was a healthy professional, a father of two. He had lots of friends, loved telling jokes, and played softball on Sundays. Then one day, at the age of 45, he suffered a stroke. He bounced back fairly quickly, with one major exception. He was no longer able to speak. The stroke damaged a specific area in the left hemisphere of his brain called Broca's area, and left him with what's known as Broca's aphasia. Broca's area is partly responsible for the ability to produce and process language, and Broca's aphasia often leaves its sufferers with some ability to understand speech, but an inability to produce intelligible words. James could understand his wife when she asked if he wanted cereal for breakfast, but he could only respond by repeating the word to, although he could still intonate as though he were speaking a whole sentence. Then, after some time in therapy, something rather unexpected happened. James regained some ability to communicate through singing. Broca's aphasia can sometimes be treated by teaching patients to sing, because singing uses a different region of the brain, one that's on the right side and that's analogous to Broca's area on the left. So after some practice, James could sing words, and he eventually relearned how to talk by teaching the right side of his brain to speak rather than sing. Whether it's a stroke affecting your speech, a tumor destroying your memory, a concussion affecting your aggression, or that fateful iron rod that shot straight through Phineas Gage's skull, a lot of what we know about how the brain works has come through studying injuries to it. And what we've learned so far is that even though it looks like a 1.4 kilogram lump of gray congealed oatmeal, the brain is made up of super specific areas that have super specific functions. You might actually say the same thing about your brain that's sometimes said about politics. Everything is local. You'll remember that our nervous system is divided into two main networks that work in harmony. The central nervous system, consisting of your amazing brain and spinal cord, and the peripheral nervous system, made up of the nerves coming out of that central nervous system. The central nervous system's main game is integrating the sensory information that the peripheral system collects from all over the body and responding to it by coordinating both conscious and unconscious activity. The sun is bright, so I'll shade my eyes. I'm hungry, so I'm calling the pizza man. The phone is ringing, maybe I'll answer it. All these sensations, thoughts, and directions processed through this two-part system. It's the brain, of course, that sorts out all that sensory information and gives orders. It also carries out your most complex functions, like thinking and feeling and remembering. Meanwhile, your spinal cord conducts two-way signals between your brain and the rest of your body, while also governing basic muscle reflexes and patterns that don't need your brain's blessing to work. This is how a chicken can still run around even if the poor thing has been decapitated. Both your spinal cord and brain are made of fragile, jelly-like nervous tissue that is extremely susceptible to injury. So all that goo is well protected by the bones of your vertebrae and cranium, as well as membrane layers or meninges, before being bathed in a cushy waterbed of clear cerebrospinal fluid. This fluid actually allows your brain to float somewhat in your skull, reducing its weight and letting it slosh around while you and your head are free to move. But even with all that extra protection, your brain is still vulnerable, and one thing that James's story taught us is that its vulnerabilities can be incredibly specific because your brain is divided into specialized regions that may or may not interact with each other to produce a given action. We can better understand this division of labor by looking at how the brain first develops into its main component parts. Inside a developing embryo, the central nervous system starts off as a humble little neural tube. Soon, the caudal, or lower end of the tube, stretches out, forming the spinal cord, while the cranial end begins to expand, divide, and enlarge into three primary brain vesicles, or interconnected chambers. This is kind of your proto-brain. We call these chambers the prosencephalon, the mesencephalon, and the rhombencephalon, or forebrain, midbrain, and hindbrain. By an embryo's 5th week of development, these main three chambers start morphing into 5 secondary vesicles that essentially form the roots of what will become your grown-up brain structures. The prosencephalon divides into 2 sections, the telencephalon and the diencephalon. The rhombencephalon forms into another pair called the metencephalon and the myelencephalon. And in between, the mesencephalon, thanks to evolution, remains undivided. The real action starts as these 5 secondary vesicles start developing into the major adult brain regions that you might be more familiar with: the brainstem, the cerebellum, the diencephalon, also known as the interbrain, and finally the cerebral hemispheres. But in order to go from a simple tube into that classic wrinkly icon we think of as the brain, each of these 5 vesicles grows in different ways. Basically, some develop a lot more than others. The least dramatic changes occur in the 3 most caudal or lower sections: the mesencephalon, the metencephalon, and the myelencephalon. They go on to form the cerebellum, which mostly helps coordinate muscular activity, and the brainstem, which plays a vital role in relaying information between the body and the higher regions of the brain. The brainstem actually has 3 main components, and I know this is getting to be a lot of vocabulary here. You have the midbrain, the pons, and the medulla oblongata. Together, they regulate many of the most basic vital involuntary functions, like keeping your heart on pace, keeping your lungs working, and controlling things like sleep and appetite and pain sensitivity and awareness. But of the 3 brainstem parts, it's your midbrain that carries out the higher level functions. Like when your eyes track a fast-moving object, or when you look behind you after hearing some sudden loud sound, it's the midbrain that receives and processes that sensory information and sends out the reflexive motor signals so you react without thinking. The midbrain also passes that data to regions like the cerebral cortex, which do the actual conscious thinking about the stimuli, like, what is that thing whizzing across the sky, or what just exploded behind me? So with the brainstem and the cerebellum covering your basic life and motor functions, you start to see somewhat more complex tasks being carried out in the next major brain structure, the diencephalon. This is where you find the thalamus, hypothalamus, epithalamus, and the mammillary bodies, which regulate things like homeostasis, alertness, and reproductive activity. Here we also find part of the limbic system, which is a center for strong emotions, like fear. This area is sometimes called the reptilian brain because we share it with some of our less philosophical animal brethren like lizards and fish. I'm not putting these guys down, but by our standards, they don't think so much as focus on the more instinctual pursuits that are ruled by the caudal regions of the brain: eat, drink, sleep, mate, stay safe. All those things are awesome, but it wasn't until the appearance of birds and mammals that some animals' brains came to be dominated by the last of the 5 vesicles, the telencephalon. During your brain's growth, the telencephalon undergoes the biggest changes of all as it develops into the most brainy part of your brain. The 2 classic walnut-looking hemispheres we collectively call the cerebrum, the cover the rest of your brain like a mushroom cap on its stalk. That's the cerebrum, not to be confused with Cerebro, which is Professor X's telepathy-enhancing device, and it is the largest region of the brain and performs the highest functions. It's made up of the wrinkled outer layer of gray matter called the cerebral cortex and the inner squishy layer of white matter beneath it. And it's the cerebrum that rules our voluntary movements and our most advanced tricks, like thinking and learning and regulating and recognizing emotions and experiencing consciousness in general. You'll remember that higher processing requires lots of synapses, which require lots of nervous tissue, so as the cerebrum grew through evolutionary time, it got more massive, but our skull didn't exactly keep up. So in order to squeeze all that material into your skull, the brain forms little creases, called gyri, and larger grooves, or sulci, giving it more folds than an origami pineapple. And although a big fissure separates the left and right hemispheres, the two halves communicate through a series of myelinated axon fibers called the corpus callosum. And each hemisphere has other smaller fissures that divide it into lobes, each with a different set of major functions. The frontal lobe, for example, governs muscle control and cognitive functions like planning for the future, concentration, and preventing socially unacceptable behaviors. In most people, this area doesn't finish developing until after the teenage years, which tells you a lot about the teenage years. Since Broca's area lives in this lobe in the left hemisphere, it also is important in language comprehension and speech. If you're enjoying a beautiful sunset, you can thank your occipital lobe at the back of your head for processing those bright visual cues. And the next time you step on a Lego, you can curse your parietal lobe, which processes the sensations of touch, pain, and pressure. Meanwhile, the temporal lobe helps sort out auditory information, including language. It contains Wernicke's area, another important region of the brain associated with the production of written and spoken language. This part of the limbic system includes your short-term memory keeper, the hippocampus, and the emotional amygdala, which controls sexual and social behavior. So if you damage the wrong part of your temporal lobe, you may never again be able to remember what you ate for lunch, or you might suddenly become a total jerk who kicks kittens and cuts in line. We could do a whole course on the finer-grained functions and consequences of malfunction and every bit of brain in your gourd, but, well, we can't do that today. And you gotta remember that when it comes to your body, no organ or system is an island. Your brain would be pretty useless if it weren't hooked up to the outside world. That's where the peripheral nervous system comes in, which we'll be spending the next few lessons exploring. Meanwhile, you learned today about the central nervous system and how important location is to brain function. We looked at how the brain develops from an unassuming neural tube into 3 primary vesicles and then 5 secondary vesicles, and finally into our complex set of 4 adult structures and their basic functions. Crash Course is now on Patreon. Thank you so much to all of our supporters on Patreon who help make Crash Course possible for themselves and for everyone else in the world through their monthly contributions. If you like Crash Course and you want to help us keep making great new videos like this one, you can check out patreon.com/crashcourse. This episode was written by Kathleen Yale, the script was edited by Blake DiPestino, and our consultant is Dr. Brandon Jackson. It was directed by Nicholas Jenkins and Michael Aranda, and our graphics team is Thought Cafe.